Hello, everyone. Welcome back. In the previous lecture, we learned that adaptive immunity relies on highly specific B cells and T cells, and that only lymphocytes capable of recognizing a particular antigen are activated through clonal selection. However, this raises an important question. If T cells are so important, how do they actually detect infection? Unlike B cells, T cells cannot simply bind free antigens floating around the body. Instead, antigens must first be processed and displayed to them. In this lecture, we'll explore antigen presentation, MHC molecules, dendritic cells, and how helper and cytotoxic T cells become activated. By the end of this module, you should be able to explain the function of MHC molecules, differentiate between MHC1 and MHC2, and describe how helper and cytotoxic T cells become activated. B cells and T cells recognize antigens in different ways. B cells can bind intact antigens directly using their B cell receptors. They can recognize proteins, carbohydrates, and other molecules on the surface of pathogens. T cells are different. They cannot recognize free antigens. Instead, T cells require other cells to process antigens and display small fragments on their surface. This means T cells are dependent on a communication system that allows cells throughout the body to report what they contain or what they have encountered. That communication system is antigen presentation. Antigen presentation begins when proteins are broken down into small peptide fragments. Cells continuously degrade proteins as part of normal cellular maintenance. If a cell becomes infected by a virus, viral proteins are also broken down into peptide fragments. These peptide fragments are loaded onto specialized proteins called major histocompatibility complex molecules, or MHC molecules. MHC molecules function like molecular display cases. They carry peptide fragments to the cell surface where they can be inspected by T cells. When a T cell encounters an MHC molecule displaying a peptide that matches its receptor, it receives information about what is occurring within or around that cell. Without MHC molecules, T cells would have no way to determine whether cells are healthy, infected, or associated with a pathogen. There are two major classes of MHC molecules. The first is MHC class 1. MHC1 is present on nearly all nucleated cells in the body. Because almost every cell can become infected by a virus, almost every cell needs a way to report its internal condition to the immune system. MHC1 displays peptides derived from proteins produced inside the cell. Under normal conditions, these peptides come from the cell's own proteins. However, if the cell is infected by virus, viral peptides will also appear on MHC1 molecules. Cytotoxic T cells, also called CD8 positive T cells or CD8 plus T cells, continuously monitor MHC1 molecules. When they detect foreign peptides displayed on MHC1, they recognize that the cell is infected and may need to be destroyed. The second major class is MHC class 2. Unlike MHC1, MHC2 is found only on specialized immune cells called antigen presenting cells. These include dendritic cells, macrophages, and B cells. Rather than displaying proteins produced inside the cell, MHC2 displays proteins that were engulfed from outside the cell. For example, a dendritic cell may engulf a bacterium, digest it, and then display bacterial peptides on MHC2 molecules. Helper T cells, also called CD4 plus or CD4 positive T cells, recognize antigens presented on MHC2. A useful way to remember the difference is that MHC1 reports what is happening inside a cell, while MHC2 reports the cell has encountered what the cell has encountered outside itself. And then just like a nerdy math way to remember is if you think of like MHC1, CD4, MHC2, CD8, so it's doubling both times. All right. Dendritic cells play one of the most important roles in the immune system because they connect innate immunity to adaptive immunity. Dendritic cells use innate immunity receptors to detect pathogens and signs of infection. They then engulf pathogens and process their antigens. After encountering a pathogen, dendritic cells migrate to nearby lymph nodes where large numbers of naive T cells are located. There, dendritic cells display antigens on MHC molecules and provide additional activation signals that help initiate adaptive immune responses. Without dendritic cells, T cells would rarely encounter the antigen they need to become activated. For this reason, dendritic cells are often described as the bridge between innate and adaptive immunity. One major outcome of antigen presentation is the activation of helper T cells. Helper T cells recognize antigens displayed on MHC2 molecules. However, antigen recognition alone is not enough. The T cell must also receive additional activation signals known as co-stimulatory signals. These extra signals help ensure that helper T cells are activated only when a genuine threat is present. Once activated, helper T cells function as coordinators of the immune response. They release signaling molecules called cytokines that influence the activity of many other immune cells. Helper T cells activate macrophages, stimulate B cells to produce antibodies, recruit additional immune cells to sites of infection, and help direct the overall immune response. Although helper T cells generally do not kill pathogens directly, they are essential because they coordinate um, many aspects of immunity. Another important outcome of antigen presentation is the activation of cytotoxic T cells. Cytotoxic T cells recognize antigens displayed on MHC1 molecules, like helper T cells. Uh, like helper T cells, they require both antigen recognition and additional activation signals before becoming fully activated. Once activated, cytotoxic T cells patrol the body, searching for infected cells displaying the same antigen. Their primary role is to eliminate cells infected by viruses and other intracellular pathogens. Instead of attacking pathogens directly, cytotoxic T cells destroy the infected host cell that are serving as the pathogen factories. When a cytotoxic T cell recognizes an infected cell displaying the appropriate antigen on MHC1, it forms a close connection with the target cell. The cytotoxic T cell then releases specialized molecules called perforin and granzymes. Perforin helps create pores in the target cell membrane. These pores allow granzymes to enter the infected cell. Once inside, granzymes activate a series of enzymes that trigger apoptosis or programmed cell death. Apoptosis is a highly controlled process that causes the infected cell to dismantle itself in an orderly manner. By inducing apoptosis, cytotoxic T cells eliminate infected cells while minimizing damage to surrounding tissues. This mechanism is especially important for controlling viral infections because viruses rely on living host cells to reproduce. So, in summary, T cells cannot recognize free antigens. Instead, antigens must first be processed and displayed on MHC molecules. MHC1 is found on most nucleated cells and presents intracellular antigens to CD8 cytotoxic T cells. MHC2 is found on antigen presenting cells and presents extracellular antigens to CD4 helper T cells. Dendritic cells serve as a critical link between the innate and adaptive immunity by capturing antigens and activating naive T cells. Helper T cells coordinate immune responses through cytokine production, while cytotoxic T cells destroy infected cells through the actions of perforin and granzymes. In the next lecture, we'll focus on B cells and antibodies and explore how the adaptive immune system neutralizes pathogens outside of cells. With that, thank you so much for attending this lecture, and I'll see you in the next one.